We will continue our discussion about this C1GalT1 knockout in the case of this pancreatic tumor mice model. So what we will observe, what we have observed in this panel, or the summary of this panel, is that we will understand is will leads to this aggressiveness of the tumors that we will see in the case of this C1GalT1 knockout mice model. So, and these studies are being compared against the mice model that has this pancreatic tumor, but this C1GalT1 is not knocked out. So, and that mice model has this KRAS mutation and TP53 mutations along. Yeah, so those mutations are there in the case of this control mice model. So initially, in order to check this aggressiveness of the tumor or the proliferation of the tumor, this Ki staining has been carried out, and we can see that in the case of this KPCC, the staining is more prominent compared to the KPC. And one more thing that we need to know about these results is that this KPC stainings are done after at a later stage, that is around 20 weeks, where this KPCC stainings are done at around 10 weeks. So this indicates that in the case of the C1GalT1 knocked out, the proliferation of the tumors are much faster, and this Ki staining is one of the biomarker that is used to detect the proliferation of the cancer or tumors within the patient, within the patients or patient samples. Yeah, and this is the quantitative results of this fluorescence image, that we can see that the KPCC has in KPCC the Ki staining is significantly higher compared to this control case of that is KPC. Later on they check the stroma, the presence of stroma in the case of presence of stroma in the tumor in different mice models. And stroma is basically the layer of tissues that is formed across the tumor. We can say a benign layer of tissue that is formed across the tumor, and they use this smooth muscle marker, that is alpha SMA, to detect this to detect the content of this, to detect the presence of the stroma. And surprisingly, they have seen that the stroma is low in the case of this KPCC mice compared to this KPC mice, and this is also validated by the quantitative results of these fluorescence images. And this indicates that the there is a less presence of the stroma in the case of these KPCC cells. And one of the rationale behind this could be that because of the high proliferation of the tumor cells, they will not be able to provide a time for the stroma formation. And these things are again validated using this collagen 1 marker, where they detect the presence of collagen 1 on both the mice models, that is on the control and all the C1GalT1 knocked out mice models, pancreatic mice models. And they can see that in the case of this KPC mice model, the collagen present is high compared to the case of this KPCC mice model. And these things are also validated using quantitative measurement of this fluorescence image, and both the results are in line, shows the same conclusion, that amount of stroma is low in the case of this KPCC mice model. And finally they check using this Cyrus red. This Cyrus res is indication of this collagen 1, is the marker for this collagen 1. And this one is the panel E, and panel F is the experiment for this smooth muscle actin presence. And this KPCC marker is also a kind of, we can say, is also the kind of indication of the stroma cells. And we can see that in the case of KPCC, the Cyrus red is low, So this indicates that this further substantiates the results from the panel C, D, E, and F, that less formation of stroma in the case of this KPCC compared to the KPC, and these things, and panel G, images in the panel G are validated using this panel H. So in order to clear the confusion, the panel C is about the smooth muscle actins. They can see that smooth muscle actins are low in the case of KPCC compared to this KPC, and when they check this smooth muscle actin using the fluorescence, and they find out the similar results, and thereafter they validated those results using this collagen measurement using Cyrus red, and they can see that the collagen formations are low in the case of KPCC. So all these results indicates that the aggressiveness of the tumors in the case of C1GalT1 knocked out, and they even lead to the less trauma formation because of the less time available for this stroma formation across the tumor cells as they are continuously proliferating. I hope you have understood how this C1GalT1 knocked out is related to this aggressiveness of the tumor, more specifically the pancreatic tumor. If you need to understand more about this role of this C1GalT1 in the case of this pancreatic cancer or PDAC, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.